Hi all. Have you ever wondered why bacteria are stained before we observe them under a microscope? Take a look at the first image on the screen. This is how bacteria appear under bright field microscope without staining. They are almost transparent and only faint outlines are visible. Because of this, it becomes very difficult to identify bacterial cells, study their shape and observe their arrangement. So now compare it with the second image. Once the bacteria are stained, they become visibly clear. Okay, some appear purple while others appear pink, and their shape, arrangement, and overall morphology become much more easier to study. So, the primary purpose of staining is to improve the visibility of bacterial cells under the microscope, and among all the staining techniques, one of the most important and widely used method in microbiology is gram staining. And gram staining is a differential staining technique developed by Hans Christian Gram in 1884. It is called the gram staining and because it was named after its inventor. Using this technique, bacteria are classified into two major groups, gram-positive bacteria and gram-negative bacteria. But here's an interesting question. Every bacterial sample is treated with the same reagents and follows exactly the same staining procedure. So why do some bacteria remain purple while others remain pink? To answer that question, let's first understand the four reagents used in gram staining. Okay, now let's go to the reagent slide. So, gram staining uses four important reagents and each one has a specific function. See, this is a gram staining kit. You can see all of the four reagents here. So, the first reagent is crystal violet. This is called as a primary stain and it is in purple in color. When crystal violet is added, all the bacterial cells, whether gram positive or gram negative, they become purple initially. Then the second reagent is grams iodine. It is called as a mordant. You know what is a mordant? A mordant is a substance that helps the stain bind more firmly to the bacterial cell wall. Instead of simply coloring the cells, iodine combines with the crystal violet to form a larger or more stable complex called the crystal violet iodine complex or CVI complex. The third reagent is alcohol or 95% ethyl alcohol. This is a decolorizer and it is most critical important reagent in which the entire procedure is dependent. Okay, this is a step that finally determines whether a bacterium will finally be a gram positive or a gram negative. The fourth and the final reagent is safranin. Safranin is a counter stain and it stains only those bacterial cells that lose the primary stain during the alcohol treatment, giving them a pink or red color. Okay, now that we understand the function of each reagent, let's go through this gram staining procedure step by step. Okay, so the procedure is simple when followed in a sequence. First, prepare a bacterial smear on a clean glass slide. Now, add crystal violet and allow it to act for 60 seconds. At this stage, every bacterial cell becomes purple. And then you have to wash it gently with water and then add grams iodine for another 60 seconds. During this step, the crystal violet it combines with the iodine to form the crystal violet iodine complex, which is larger and more difficult to remove from the cell. Now the cells are still in purple at this point. Now comes the most important step in gram staining. Now we will have to add the alcohol for 5 to 10 seconds only. Timing is extremely important. If alcohol is applied for too long, it may lead to incorrect results because even gram-positive bacteria can lose the stain. Then, after decolorization, Rinse the slide and apply safranin for approximately 45 seconds. And finally, wash the slide with water, dry it and observe it under the microscope. You will notice that some bacteria remain purple while others remain pink. Now, but why does this happen? The answer lies in the structure of the bacterial cell wall. Gram-positive bacteria, they possess thick peptidoglycan layer, and in contrast, gram-negative bacteria have thin peptidoglycan layer and an additional outer lipid membrane. Now, this structural difference is the reason both groups respond differently during the alcohol treatment. Now, let's see exactly how that happens. Okay, now the most important step is decolorization. Okay, here let's begin with the gram-negative bacteria. When alcohol is added, it dissolves the outer lipid membrane because lipids are soluble in alcohol. Remember that, right? So as the outer membrane breaks down, tiny pores are formed in the thin peptidoglycan layer. Through these pores, the crystal violet iodine complex escapes from the cell, and as a result, gram-negative bacteria become completely colorless after the alcohol treatment. Through these pores, the crystal violet iodine complex escapes from the cell. As a result, gram-negative bacteria become completely colorless after the alcohol treatment. When safranin is added afterwards, these colorless cells absorb the pink dye and appear pink under the microscope. Now let's look at the gram-positive bacteria. See, these bacteria have much thicker peptidoglycan layer. When alcohol is applied, this thick layer becomes dehydrated and shrinks. As it shrinks, the peptidoglycan network becomes tighter, trapping the crystal violet iodine complex inside the cell. Now you can see in this picture, right? Since the purple stain remains trapped, the bacteria continue to appear purple. Even after adding safranin, the strong purple color masks the pink counterstain. Okay, therefore, gram-positive bacteria remain purple. A simple way to remember this concept is thick wall traps the purple, thin wall loses the purple. Okay, now let's look at few common examples. Examples of gram-positive bacteria include Staphylococcus aureus, Streptococcus pyogenes, Bacillus subtilis. Now, the examples of gram-negative bacteria includes Escherichia coli or E. coli, Pseudomonas aeruginosa, Salmonella typhi. Now that we understand the mechanism, let's see why gram staining is so important in microbiology. See, gram staining is one of the most frequently performed techniques in microbiology because it provides valuable information within a very short time. At first, it helps in the primary classification of bacteria into gram-positive or gram-negative groups. Second, it provides rapid preliminary identification, like when a clinical sample reaches the laboratory. Gram staining can give an early indication of the type of bacteria present even before the culture results are available. Third, it plays an important role in guiding antibiotic therapy. Gram-positive and gram-negative bacteria often respond differently to antibiotics because of their differences in their cell wall structure. Although the antibiotic selection ultimately depends on factors such as bacterial species and antibiotic susceptibility testing, gram staining provides an important first clue that helps clinicians choose an initial treatment while waiting for a definitive laboratory results. Okay, finally, gram staining is routinely in clinical laboratories, microbiology research, teaching laboratories, diagnostic microbiology. It remains one of the most fundamental techniques every microbiology student should understand. So to summarize, gram staining is a differential staining technique that separates bacteria to gram negative or gram positive based on differences in their cell wall structure. So the key step is decolorization with alcohol and understanding the structure of the bacterial cell wall. I hope this explanation helped you to understand the concept clearly. So in next video we'll move from bacteria to fungi and begin a new topic, morphology of mold and yeast, and where we learn how molds and yeast differ in structure, growth, appearance and all. So thank you for watching and see you in the next class.